Halting a runaway biological process, such as high cholesterol or a bacterial infection, requires medicine to intervene at the molecular level without damaging healthy cells. Success depends on the precise interception of enzymes. Looking at this simplified model, an enzyme functions as a specific docking station. The active site is shaped to accept only a substrate with the exact geometric complement required for a reaction. Inhibition occurs when a foreign molecule occupies the site instead. When the inhibitor is docked, the normal substrate deflects away, and the reaction cannot proceed. This microscopic interaction governs a vast range of pharmaceutical designs. From simple pain relief to complex antibiotics, many medicines work by providing these decoy molecules to stop specific enzymatic reactions. Mastering the mechanics of these molecular blocks is the core of modern drug design and disease control. One primary method is competitive inhibition. As the name suggests, a red imposter molecule mimics the shape of the normal substrate, attempting to secure the same docking point. Both molecules are now vying for access to the same physical location. Because the enzyme can only hold one molecule at a time, the inhibitor physically blocks the substrate's path. This physical competition can be resolved through sheer numbers. By overwhelming the system with an excess of normal substrate, the probability of the substrate reaching the active site first increases, eventually restoring the reaction rate. Statin drugs operate on this principle. They mimic the natural substrate to competitively block HMG-CoA reductase, the enzyme responsible for synthesizing cholesterol in the liver. In this scenario, the active site is controlled by whichever molecule achieves the highest concentration. Non-competitive inhibition uses a different tactic. These inhibitors ignore the active site entirely, targeting a secondary location on the enzyme known as the allosteric site. When an inhibitor docks into the allosteric site, it alters the enzyme's three-dimensional structure. This change in conformation distorts the active site, making it impossible for the substrate to fit. Because the lock itself is now the wrong shape, numbers no longer matter. Even with hundreds of substrates present, they cannot bind to a deformed active site, making this type of inhibition immune to substrate concentration. Think about these two mechanisms. Which one will place a permanent ceiling on the maximum speed at which a biological system can operate? Non-competitive inhibition effectively removes enzymes from the total pool, neutralizing their function from the inside out, regardless of how much substrate is available. Biochemists measure these interactions using two key metrics: Vmax, the maximum speed of the reaction, and Km, the substrate concentration needed to reach half of that maximum speed. In competitive inhibition, Vmax stays the same because infinite substrate can still displace the inhibitor to reach top speed. However, because the inhibitor makes it harder for the enzyme to find a substrate at lower levels, you need more substrate to reach that half maximum speed. Therefore, the Km increases. With non-competitive inhibition, Vmax drops because the total number of functioning enzymes has been reduced. Yet the remaining enzymes that aren't inhibited still bind to substrates just as easily as before, so the Km remains unchanged. These kinetic shifts provide a mathematical fingerprint, allowing researchers to identify exactly how a substance is interfering with a metabolic pathway. For quick recall during study, competitive means a climbing Km, while non-competitive means Vmax takes a nosedive. To summarize, competitive involves fighting for the same site, where the substrate can still win. Non-competitive involves a separate site that changes the enzyme shape, meaning the substrate cannot win. A more dangerous complication occurs with lethal nerve agents like sarin. These molecules form permanent covalent bonds that fuse to the active site, causing an irreversible failure of the enzyme. Based on the kinetic patterns we've discussed, how would you classify this irreversible threat? Share your reasoning in the comments. Understanding these specific molecular interactions is essential for both medical innovation and defense against toxins.